Good afternoon, ladies and gentlemen, and welcome to the Destiny Pharma PLC Capital Markets Day. Throughout this recorded presentation, attendees online will be in listen-only mode. Questions are encouraged. They can be submitted at any time using the Q&A tab just situated on the right-hand corner of your screen. Just simply type in your questions at any time and press send. Given the attendance on today's call, the company will not be able to answer all the questions submitted today. However, the company can review all questions submitted today and will publish those responses on the Investor Meet Company platform. Before we begin, we'd like to submit the following poll, and if you could give that your kind attention, I'm sure the company will be most grateful. And you'll also find an agenda for today's Capital Markets Day in the handout section on the right-hand side of your screen. I'd now like to hand over to the presenting team from Destiny Pharma. Good afternoon.
Yeah, good afternoon and good morning to everyone. My name is Yuri Martina. I'm the Chief Officer of Destiny Pharma, and I'm very pleased to welcome you all to this event. We will be speaking about the XF pipeline update and how exciting the XF pipeline is, and we'd like to share with you this excitement. We have a series of presenters from Destiny Pharma. We have Chris Tovey, our Chief Executive Officer, and William Love, our Chief Scientific Officer, and then we have a series of key opinion leaders in the infectious disease, experts, or surgeons. Unfortunately, one of the experts could not attend today, Dr. Ragbir Kaka. He has been called in an emergency surgeries due to the known strikes in the UK and perhaps a surgical site infection, which will be one of the topics that we will be discussing today. Without further delay, I hand over to Chris Tovey, who will introduce the agenda and the day.
Thank you very much, Yuri. And next slide, please. So obviously a big welcome today to this session from wherever you're joining us. We've got a very healthy audience, which you can't see if you're on camera, but I know we've got a lot of people online as well. But just wanted to welcome you all on obviously a very exciting day for Destiny. I think most of you will have seen the data from this morning, something we're amazingly proud of and excited by. And Bill will talk about that later. Dr. Love will talk about that data later. So it's really down to me to introduce the day and the session, and to talk to really Destiny Pharma, and to give you a sense of why, well, partly why I joined Destiny, but also just why we think this XF73 nasal is so exciting. It's going to make such a big difference for patients. Agenda, you can see we have a number of speakers, and they are going to work their way through surgical site infections from all angles. Some brilliant speakers today, and we will also, Bill will cover some real scientific detail on XF73 itself, which I think some of that information a number of you will know already, but there's one or two pieces of data and information there you may not be so clear about. And we constantly get asked about the comparison with mupirocin, and Bill's going to talk about that in as clearer terms as we possibly can. So next slide, please. And it falls to me to welcome our speakers today. We've got brilliant speakers. And first and foremost, we have Professor Mark Wilcox. And Mark is an expert in infectious diseases and currently serves in several substantive roles, including consultant microbiologist and head of research and development in microbiology at the Leeds Teaching Hospitals. He's also National Clinical Director of AMR for NHS Improvement England. And you can see why he's such a good speaker for us, and a lead on Clostridium difficile for Public Health England. Obviously today we're going to focus on XF73, not on NTCDN3. He's served in multiple advisory roles, including to UK SAGE, I think an acronym that we all got used to a couple of years ago. And he's also a member of the UK NHS Antimicrobial Resistance Programme Board. So Mark, brilliant speaker, and is going to bring some tremendous insights for us all today. Our next speaker is Dr. Alex Merickley. And Alex is joining us from the States today, so reasonably early for him in the morning. He's an associate professor of plastic surgery at the University of Texas MD Anderson Cancer Center. He is interested in all aspects of reconstructive surgery with particular attention to breast, head and neck, and trunk reconstruction. He's dedicated to surgical research and education, and has authored or co-authored over 70 peer-reviewed articles and book chapters on various plastic surgery topics. He's lectured nationally and internationally on his research, so he's a fabulous speaker for us to have on this call. Actually, an additional thing about Alex, which I think is extremely important when you're thinking about XF73 nasal, is he's also published a number of articles detailing the cost effectiveness of several different plastic surgery procedures and technology. And I think we all know that that cost effectiveness aspect of XF73 nasal is going to be extremely important to its successful commercialization. The next speaker I'd like to introduce is Professor Richard Proctor. Richard's a Professor Emeritus at Department of Medical Microbiology, Immunology, and Professor Emeritus of Department of Medicine at the University of Wisconsin School of Medicine and Public Health, Madison, Wisconsin. So somebody else from the other side of the Atlantic today. He's also served as Global Director of Infectious Diseases for Merck Research for antibiotics and antifungals. Most of his recent work has focused upon aspects of staphylococcal infections, and he spent more than 2 decades studying macrophage activation, basic immunology, and sepsis. He's a leading expert on bacterial pathogenesis of infectious disease. And like some of our other speakers, he serves on a number of panels addressing emerging antibiotic resistance. So delighted to welcome Professor Proctor as well today. So next slide, please, Mark. I think everybody probably who's interested in AMR has seen the sort of statistics that you see on this slide. I think the World Health Organization is pretty clear that antimicrobial resistance is a global threat, and something that many organizations, governments, supranational organizations are taking very, very seriously. And that is the canvas on which Destiny Pharma finds itself. Next slide, please. So who are Destiny Pharma? Who are we? Well, we're a company that's developing novel medicines that prevent serious infections. 2 programs in particular that those of you who are familiar with Destiny will have heard of. Our NTCDM3 program, which is operating in the C. diff space. And importantly for today, XF73 nasal, which is for the prevention of post-surgical Staph aureus infections. We believe that that product is positioned to target an up to $2 billion market in the US. And we obviously also believe, and we're hoping that at the end of the day, we reinforce that belief in your minds, that it's an incredibly strongly positioned product, both against standard of care and actually against products in general. Destiny Pharma, next slide, please, Mark. So just now to home in on talking about surgical site infections. You're going to hear more from experts and you're going to hear from me on this, but I think the starter here is, they are a major cause of patient morbidity and mortality. SSI accounts for about 20% of all hospital-acquired infections and is associated with a sort of a 2 to 11-fold increase in the risk of mortality, with 75% of SSI-associated deaths directly attributable to the SSI, surgical site infection. These are serious things, very, very serious things. And I think that, XF73 nasal, very clearly a serious medicine. They're associated with significant increased healthcare costs, and they represent both a burden to patients and a significant burden to families and to the healthcare system. Staph aureus, including important antibiotic-resistant strains such as MRSA, something we've been hearing about now for 2, 3, 4 decades, are a common cause of nosocomial infections in hospitals worldwide. So this is a common issue. This is a serious issue and a common issue. The WHO has identified Staph aureus as a major health concern due to the growing threat of AMR. And people have different numbers on this, but the reality is, unless action is taken, significant numbers, 10 million in this particular reference, 10 million AMR deaths are estimated each year by 2050, with a cumulative cost of, I can't quite recognize this number, 100 trillion US dollars. This is a serious impact. We've all just lived through COVID. Nobody ever thought that was going to happen. And it did. So, I think people really do see this as a serious issue that is heading towards us. Next slide, please, Mark. Many drugs that are introduced these days increase the cost of the healthcare system. One of the brilliant things we believe about XF73 nasal is we actually think it's likely to be a drug that helps the healthcare system manage costs, particularly in the US. That Staph aureus SSI carry up to $165,000 incremental cost per patient. And there are millions, tens of millions of surgeries at risk every year of those, of that consequence. And the way the US system works, obviously a surgical site infection attracts increased costs. But also many hospitals are tracked by CMS on quality metrics and are also tracked by payers around quality metrics and penalties. So in terms of the incentive for the hospital system, the payer system in countries like the US and in the UK and the rest of the major markets in Europe, there are some very significant tailwinds encouraging the use of prophylaxis to reduce surgical site infections. Next slide, please, Mark. So what we've seen develop over the last 20 or 30 years is one particular intervention that helps reduce surgical site infections, and that is nasal decolonization. When I started working with antibiotics in the late 80s, nasal decolonization was still in its very early genesis, and the idea of anything being used up your nose to stop a Staph aureus-based infection was still an incredibly new concept. Now, as we look, and I'll show a slide in a minute, as we look now at the situation, you have got a wide penetration now of nasal swabs being conducted and people being treated with a nasal decolonization agent. And you've also got, even in some situations, and increasingly so, you've got physicians universally decolonizing. Basically, they don't take a swab. They just believe it's a lot easier and a lot more efficient just to decolonize a patient with a nasal prophylactic. And this isn't just a US phenomenon. This is going on all over the world. So you can see many different healthcare systems, UK are taking very much this prophylactic action because they can see the consequences of out of control surgical site infections and the opportunity to avoid them with nasal decolonization. Next slide, please, Mark. Just some numbers from some recent research we've done. And that research was very much looking at the more at-risk surgeries, which are still a significant proportion of the total number of surgeries in the US or Europe. And what we saw in the US was that up to 40% of surgeons are actually screening, just generally across surgical procedures. And then in the more serious end, we find surgeons, nearly 60% of them, conducting universal decolonization. So basically, without taking a swab, they are choosing to put an agent up somebody's nose to decolonize the Staph aureus that's being carried there. And I just think that is such a huge step forward from where we were just a decade, 2 decades ago, and is a really important situation in which then we would find XF73 nasal coming into. Rather than market development, this is about market penetration and taking share away from a pre-existing agent that's already being used widely. Next slide, please. Now, Bill's going to do a much better job than I am talking about how amazing XF73 is. But I just couldn't resist pulling out a few bullets for everybody, just because this is the main reason I joined Destiny. You know, I'm absolutely 100% behind the idea this is going to be a very successful commercial product, and that this is going to be a product that makes a massive difference for patients and for the healthcare system. So it's a new class of antimicrobial drugs. And we know, because we've bounced this clinical profile off physicians in major markets, particularly the US, we know it's got a compelling clinical profile. Phase 2 results reinforces that. It's got a broad spectrum of activity across gram-positive bacteria. It's antibiotic sparing. You know, fewer infections means less times that you have to use an antibiotic. It's got a remarkably fast bactericidal mechanism, and very much we believe that helps produce that reduction in the threat of resistance. And we believe, because it's of its relatively simple manufacturing processes that we associated with it, it's going to enable us to have strong pharma margins on the product. So, you know, this is a really, really very exciting product in a very difficult area, and an important one for hospitals, for physicians, and for patients. Next slide, please, Mark. I showed this slide recently, so for those of you that have seen it already, I apologize, but I'm just going to briefly talk about some recent market research we did in the US and in the major European markets. You know, what we did was we clarified that high-risk surgeries were absolutely relevant target opportunities for us. And when you talk to the physicians that are conducting those high-risk surgeries, really across the board now, we are seeing an acceptance of the role of nasal carriage in creating infections, causing infections, and an acceptance of the need for decolonization strategies. Physicians really positive about the XF73 profile. You know, speed of action, and you know, the paper this morning I think reinforced it, but that tremendous spectrum of Staph aureus activity, and the reduced resistance risk from using this agent. The 1-day dosing window, a lot simpler than standard of care, means greater flexibility in the system. Greater flexibility to the point where if somebody comes in as emergency, you can at least attempt to get some reasonable nasal decolonization before somebody's operated on, but also dosing the day before a surgical procedure is much more likely to achieve compliance than 5 days before. The ability to change patients at a day's notice and still achieve effective nasal decolonization with XF73 nasal was believed to be a really important opportunity. And always people want to know what the payers think. And the US payer feedback suggested quite strongly that XF73, in the context of the overall costs of procedures and the overall context of the risk cost, if it goes wrong, that XF73 nasal could achieve a really strong price versus standard of care. And you know, when you added all of that up, when we put it into our model and we started looking at the patient potential and the opportunity, we see a very significant commercial opportunity, potentially up to $2 billion in the US peak sales. So very exciting commercially. Next slide, please, Mark. So what are we doing at the moment? I think very well rehearsed a few weeks ago. Obviously, the team's job at the moment is to find a a great deal for XF73 nasal. Find a brilliant partner who's going to be able to make the most of this exciting asset. So that's our absolute focus as a management team. But less about us now and more about the rest of the session and some of our very learned speakers who are going to give you a brilliant tour of everything that's surgical site infections. Next slide, Mark.
Yeah. So thank you very much, Chris. And the next section with in the next section with Mark, Richard, Alex will give you a lot of numbers and the entity of the surgical site infection problem and the consequences from a healthcare perspective or a surgeon perspective. Before we go there, actually, we have very recently collected and recorded an interview from a UK patient. So someone who lives here in UK and has gone through, is a young patient, he has gone through a routine surgery, a routine knee surgery, and he had to deal with surgical site infections. With a surgical site infection, with all the consequences, and you will see on the video from his statement that this consequence have protracted more than 6 months, and he is still undergoing procedures to remedy the problem. So if we can play the interview.
I tore my ACL, and that was the only thing which was torn. That happened in December, and then I proceeded to get the ACL surgery via hamstring graft on end of Feb, and all was going really well, straightforward surgery. And then I had my surgery on the Thursday, and then it was a Saturday 2 weeks later, so it was almost 16 days after. I was feeling really good. I went to sleep, and then overnight, just had loads of pain in my knee. And bear in mind, my recovery was going really well. Like, I had very little inflammation, and then all of a sudden, I had pain overnight, and I was like, oh, maybe I overdid some of my physio exercises. I was speaking to the hospital, and they were like, come in tomorrow on Monday or speak to us. So then I went in the evening, and they were like, you're spiking a really high temperature. Suspected, in fact, we need to operate on you. And then they went in. They didn't find like a pussy liquid, but they found like a murky, like I think they said dark murky liquid. At that stage, it was quite hopeful that everything was going to be okay. Anyway, they gave me antibiotics, and the hope was that I was going to get better. So they planned another clean out on the Thursday, and they had hoped that I would have got a bit better, but my range of movement still wasn't very good. So we did another clean out of the knee, maintaining the graft, in hope that I'd be getting better. In between that period of that first clean out and the second clean out, I was still having those antibiotics were not making me feel good, and then B, I was still spiking like a little bit of temperature change and fever. So then the plan was to meet again the following week and just see how everything is. And I got a little bit better, but not drastically better. My movement still wasn't good. They tested the liquid inside my knee. They sent that off to the lab, and nothing was coming up. Nothing was showing as there's an infection. It took about 10 days for something to show. So that's when we knew it was that Staphylococcus. I then got my ACL removed, then went on intravenous antibiotics, and they kind of knew what to target it with because they knew what the bacteria was. Then we planned another washout 4 days later. Then it was a little bit uncertain whether I was going to need another washout, but fortunately I just ended up doing 12 days in hospital, and then I did 2 weeks at home of doing the self-inject antibiotics, and then I got the all clear that I don't have the infection, and then I could just proceed with getting stronger in physio. So I have to wait 6 months for my holes to be okay to try again. obviously it was a prolonged period. So it was had the surgery and then recovering from the initial surgery, and then the infection came 16 days later. Then it was almost 2 weeks of that in-between stage of not knowing what's going on, doing those 2 initial washouts. So that was another 2 weeks. And then I was a full straight 12 days in hospital. So in total, it was around 14 night stays in hospital. And I was off work for about 2 months. Yeah, it was pretty crazy. It was like, you're in survival mode. So you're not really thinking of anything else and just getting better. And I think it was really hard not knowing what was going on when I was feeling suddenly really ill. My knee was really hurting. And I think the hardest thing was I did the washouts and then my knee would get so swollen because they've gone in. And then you've got to start, like it's almost like you've got to start again. But like there's little exercises, reducing the inflammation. So I had that 5 times for like the surgery, the 2 washouts, then the removal, and then the last washout. And after the last washout, like my knee was so swollen. I was just in so much pain. So fortunately, I have my own business, and luckily everything ran smoothly. There could have been a time where I was really needed, and I wouldn't have been able to do anything.
Given your experience, how do you feel about going back in?
Yeah, I'm a bit nervous, if I'm honest, but I'm going to take the precautions as you can. And ultimately, I want to be playing sport and the things I like doing and getting a stable knee. So it's just something I've got to do. I'm young and I've got to go through this in order to do the things that I want to do. A bit nervous, but I'm hoping that the precautions taken. So I think I'm going on a week of antibiotics during the surgery. So yeah, that should hopefully that covers things.
Yeah, and I would like to underline that this was a young, healthy individual, and nevertheless went through a lot of trouble, which is still under resolution. In other serious cases with the patients who are an older age, this can lead to amputation or even a higher comorbidity. So we now hand over to Professor Mark Wilcox, who will speak about surgical site infections and will frame the problem from an infectious disease perspective. Mark, to you.
Thank you, Yuri. Sorry for the pause. I was just searching for the mute button. Thank you, everybody, for being interested enough to listen to what I have to say. You've heard my introduction, and thank you for those kind words. So over the course of the next 15-20 minutes, I really want to try and emphasize a few of the points you've heard already and build on some of those from an infectious disease perspective. These are, to start with, some US data which build on some of the figures you've already heard, but these are the most contemporaneous data available via CDC and the infection surveillance system that exists for surgical site infection in the US. And essentially, it's not good news from a staphylococcal perspective, because both MRSA and MSSA surgical site infections have become more common in the most recent data set rather than less common. I'm not going to read the numbers in these bullets here, but you can see the relative risk increase by comparing those percentages. And while MSSA remains lower than pre-pandemic levels, MRSA didn't. And I've purposely put the last bullet point of these 4 in smaller font because you might think, well, what's MRSA, bacteremia or bloodstream infection got to do with surgical site infections? Well, a proportion of these bloodstream infections represents spillover, literally spillover of bugs in the wound, typically deep in the wound, surgical wound, spilling into the bloodstream and causing what can be a life-threatening infection. Now, it's only a proportion of surgical site infections responsible for these, but the fact that MRSA surgical site infections have increased, and the fact that MRSA bloodstream infections have increased, suggests that MRSA is becoming a bigger problem, not a lesser problem. And that's despite many years, decades of highlighting this organism as infectious disease public enemy number one. So that was US data. This is UK, specifically England data. And it's simply looking at those bloodstream infections again, the bacteremias that I've just referred to at the end of the previous slide. So having targeted MRSA on the left, MRSA bloodstream infections as public enemy, ID enemy number one in the UK, we saw an over 80% reduction in those infections. So we all pat ourselves on the back and say, look how well we did for those that 10, 15 years of effort to control MRSA bloodstream infections. And yet we are not too vociferous at publicising that we've made no difference to MSSA bloodstream infections. In fact, as you can see from the graph, they're getting worse numerically. The rates are increasing. So some of that, and I'll come back to this point later, is that we are targeting MRSA and not targeting MSSA in terms of our interventions in some cases. And here's the money figures, and you've already heard some of these data. But essentially, surgical site infection is expensive. Indeed, it's the most costly healthcare associated infection. And the numbers driven, the cost in dollars here is driven primarily, typically at least 80, if not 90% of these costs is driven by the increased hospital length of stay rather than the drug costs at trying to treat these infections. But when these infections do occur, as we've just heard, they are expensive in dollars, expensive in misery terms for the affected individual, expensive in reputational terms, potentially for the institution and for the surgeon. So, these slides, this slide illustrates England surveillance data of surgical site infection. This is a voluntary scheme for most of the surgical categories on here, except for the orthopedic categories, which are the ones coloured in orange, where all NHS institutions have to partake in this surveillance scheme. The detail on here doesn't matter so much, but the reason I have colour coded 3 surgical types in red or pink and 3 in orange are to illustrate the extremes of surgical site infection risk, the rates of infection. So you can see at the bottom of the table, as in the league table of risk, i.e. the highest risk, are the bile duct, pancreatic surgery, cholecystectomy, and large bowel surgery. And the rates of infection there, about 10 to 15%, are an order of magnitude at least, so at least 10-fold greater than the rates of infection you see highlighted in orthopedic surgery, hip replacement, knee replacement, repair of neck of femur. And you might think, therefore, remembering back to some of the comments earlier, that if you were going to target high risk surgery for infection, you might think, well, I won't bother with hip replacement, knee replacement, because, you know, tenfold at least less risk of surgical site infection. But of course, the consequences of having an infected hip prosthesis or knee prosthesis are extremely dramatic, and often will lead, more often than not indeed, will lead to the loss of that prosthesis, very high cost, and morbidity, as we've heard already. The last thing I want to point out from this slide is that if we compare the risk of infection in elective surgery, so for example, hip and knee replacement surgery, here we see 0.4, 0.5%. Compare that with acute surgery, repair of neck of femur, typically the old lady who falls over, fractures the neck of a femur, and a prosthesis often will need to be inserted. You'll see that the infection risk at least doubles. So by and large, acute surgery, higher SSI risk compared with elective surgery. And these data are showing the same rates, but showing the spread of rates across different institutions. And one way of interpreting this is that there are some institutions are much better at controlling surgical site infection risk than others. However, it's equally possible that the reason that you might be high up in surgical site infection risk is because of the type of large bowel surgery that you are performing. For example, if you're taking lots of gunshot wounds into the abdomen where and repairing those surgically, that they would be associated, because acute surgery, higher risk, higher infection rate, and so on. So necessarily being higher up this league table, this particular league table, doesn't necessarily mean poorer performing. I've got two slides. There's this one. I'm going to flick very quickly to the next one because they're similar, and what they do is illustrate the overall burden of different microorganisms causing surgical site infections. The first slide was for the superficial surgical site infections, and this slide for the more serious, certainly much more serious to treat and eradicate, the deeper surgical site infections. And you can see at a glance that Enterobacteriaceae, so gram-negative bacteria, E. coli and pals, Klebsiella and so on, are the most common overall. But that's because certain of that infection, high infection rate we saw in certain surgical large bowel surgery types, and so these organisms dominate when you list all the organisms from the most common to the least common. In terms of what we're talking about today, though, Staph aureus, the solid red line here, remains a persistent threat. Ignore that slope. I'm not sure if you can see my cursor, possibly not. But that slope from 2020 through 2021, that's the pandemic effect. Those data are unreliable. So the point here is that Staph aureus, despite years of targeting it in different guises, it still remains a very, very key organism in the context of surgical site infection. And outside of large bowel surgery, it's probably the most dominant surgical site infection pathogen. And these are data which I'm not going to spend any time on, just illustrating these are surgical site infection according to why were you having your joint replacement. And the only reason I really put this slide up is to show you the data for if you look at the bottom half of the table, which are the infection rates in that SSI risk column associated with revision surgery, and look at that 2.5% risk of infection associated with fracture, which leading to revision surgery, much higher, about 6-fold higher than the risk of infection for primary surgery. My point here is we're going to be seeing, of course, more and more revision orthopedic surgery, because of all those prostheses that were done 10 years ago, 15, 20 years ago, when this discipline really blossomed, and the prostheses are going to start wearing out. So infection risk in orthopedic surgery may increase because of greater requirement for revision surgery. Just a simple bit of etiology of what's the origin of these infections. Well, the great majority of the infections are caused by bugs that reside in or on the patient, him or herself, hence the box around endogenous on the left. Yes, you can acquire a surgical site infection via bugs you pick up in the OR, or the operating room, the surgical suite, but they are much less common than infections caused by bacteria that belong actually to the patient themselves. Now, that's bacteria that they may have had for years, or they may have only had for days or months. And you can see that Staphylococci, and Staph aureus in particular, is listed as the most common for both endogenous and exogenous infection. I'll come back to that point shortly in another a really important data set. Before we get there, this is a really authoritative, well-written review that was published this year, start of this year, in JAMA, and it highlights the 6 key interventions that we know are available to reduce the risk of surgical site infection. And in the middle, in bold text, you can see decolonization with intranasal anti-staphylococcal agents, usually given alongside topically trying to decolonize the patient as well from their skin. So rather than simply targeting the nose, probably the most important carriage site for Staph aureus, you target the nose and the skin. So you pick up the armpits and the groin, which are the other risk areas, by asking the patient to bathe or shower using typically a chlorhexidine type antiseptic. So the point here is that the medical community, medical and surgical community, recognize the evidence that exists for the interventions that are available. What differs is how people apply 1, 2, 3, or all 6 of those. Do they apply them all the time to everybody, or some of the time to some of the patients? Now, in getting to come back to that point, can I just reset and point out this study? This was the landmark study in the story of how to address nasal colonization with Staph aureus and how to reduce infection risk. And it was carried out in the Netherlands almost 15 years ago. And this was the New England Journal publication. You can see on the left-hand side how when you follow the patients on the blue given placebo, the patients on the green line were given nasal decolonization along with chlorhexidine as well. And look at the huge difference. That picture tells 1,000 words, the huge difference in risk of subsequent staphylococcal infection. Now, with some irony, this study, as I've said, was performed in the Netherlands. the carriage rate of MRSA in the Netherlands then and now is much lower than in many other European, North American countries. 10 to 100-fold less common to carry MRSA. And yet these authors saw fit to perform, as I've said, what turned out to be, to this day, the landmark study in this area. And indeed, not a single MRSA was isolated from the study patients. So those results I showed you, that huge difference in infection risk between placebo recipients and mupirocin recipients, was driven solely by MSSA risk of infection, not MRSA. And you can see how those different infection sites fell towards the bottom half of this table. And you can see that the great majority of the infections that occurred that were prevented were indeed surgical site infections. And as I labour, they were MSSA infections. And you can see endogenous versus exogenous, 3 to 4-fold more endogenous infections. The bug came from the patient rather than from the OR. So, faced with that information, what simplistically are our options for preventing, for giving prophylaxis in the context of Staph aureus in patients undergoing surgery? Well, at one extreme, we could screen out and find out who's carrying MRSA and give prophylaxis. If we do that, of course, we're ignoring those who've got MSSA. So we could screen and give prophylaxis to anyone who's got Staph aureus. Alternatively, we can use some other way of targeting patients, whether it's the surgical type that they're about to undergo, and or whether they're high risk because of their own characteristics. If they're diabetic, if they're seriously morbidly obese, for example, or have other chronic illnesses, they all increase your risk of infection. And you can see that the arguments for and against these different approaches remain controversial to this day. But I suggest that we will move more and more towards prophylaxing more and more people at risk. And I try to highlight that with the orthopedic prosthetic surgical types, where the risk of infection is much, much lower compared with the risk of infection in large bowel surgery. But the consequences of infection, if you are one of those relatively few to get infected, are extremely dramatic and costly. Also, remember, how can we forget, the litigious society in which we live, and that's only going to go in one direction. And patients will increasingly, and some might say correctly, question if they develop an infection caused by Staph aureus. Just remind me why you didn't give me some prophylaxis directed against Staph aureus, because here's the evidence that it works. I just want to close by showing some data, and you can see the reference at the bottom here stems from early, quite a few years ago. Sorry, it's cut off on my slide, so I can't see the actual year. Can I just pause very briefly and say that at 1 to 2 this afternoon, that's UK time, so just before this meeting started, I got an email telling me that the second author on this study, which I was the lead of, has just died. And so I just wanted to acknowledge that point. Terrible news that I received. But we carried out this study where I was the infection control doctor in Leeds at this time, and I convinced the orthopedic surgeons that rather than going through that rigmarole of screening individuals, finding out who is MRSA positive, and then giving them prophylaxis, which has hits and misses, errors occur and so on, we decided, because we had a high surgical site infection rate, we decided to use empirical mupirocin-based nasal prophylaxis with chlorhexidine. And so if you were in this high risk group, which was anybody getting a prosthetic implant, typically a hip or knee prosthesis, we started using this procedure. And so we simply compared the infection rate, surgical site infection rate before period A with after periods B and C, the introduction of that intervention. And you can see from the third bullet point down, there was a approximately 7-fold reduction in MRSA surgical site infections once we introduced this intervention. And in fact, of the few infections that did occur after the intervention, they primarily occurred in people who did not, for whatever reason, get the prescribed intervention. Here's the picture that tells 1,000 words. The green bars much taller, the infections much taller prior to the intervention, which occurred in spring of 1999. This was so successful that when national policy in the UK, when MRSA became public health enemy number one, when that dictated that we should be screening individuals and prophylaxing those who are MRSA positive, we could not convince our surgeons locally to do that. They were so wedded to these data of empirical prophylaxis, which of course has its risks because you're giving more people the drug, mupirocin, and so resistance is an issue. And we did follow individuals here and we looked to see how common resistance was. We didn't have a huge resistance problem, but this was a relatively short-term follow-up. And these are also quite small numbers. But I just wanted to highlight this study towards the bottom, the bullet points, the 4 bullet points towards the bottom of this slide. This has been published only a couple of months ago. And you can see it's data from one centre. Look at the title of their publication. Is it time for a change in MRSA decolonisation protocols? And what they're not meaning, is it a time to stop doing MRSA decolonisation? They actually recommend on the back of these really high mupirocin resistance rates that they should switch to using a different agent, povidone iodine, for nasal decolonisation. This very week in JAMA, a randomized control trial has just been performed in the intensive care unit situation, comparing mupirocin with povidone iodine. Povidone iodine inferior, statistically inferior to mupirocin. Of course, the problem here is the threat of resistance. And just the last couple of things to say about resistance is that it's more common, mupirocin resistance, in MRSA than MSSA. That appears to be driven by particular resistant clones of MRSA, and it's particularly problematic in people in whom you have to give mupirocin to repeatedly, either because of repeated surgery, or, for example, if they're a hemodialysis patient, where often these are given mupirocin prophylaxis to prevent them getting infections of their hemodialysis catheter. So, to conclude, SSSIs vary markedly in frequency according to the surgery type, and I've laboured that point. But they cost a lot and they are burdensome to the healthcare and to patients. And they're still far too prevalent, despite many years of targeting these organisms and increasing knowledge about how we can reduce risk. And my last point is that resistance is an inevitability, regardless of which antibiotic bug combination you want to talk about. We know mupirocin resistance is generated by increased use of mupirocin. And so it's inevitable that increased reliance on mupirocin will generate more resistance. Thank you for listening.
Thank you, Mark. We have on the agenda a brief break. Originally, it was scheduled for 15 minutes. I would say we reduce it to 10 minutes and we reconvene at 3:05. Right? Okay. Thank you.
Perfect. Just to say the meeting will recommence at 3:05. Thank you. Ladies and gentlemen, just to say the company have just taken a short break and the meeting will recommence at 3:05. Thank you. Ladies and gentlemen, just to say that the company has just taken a short break and the meeting will recommence at 3:05.
Thank you everyone for being punctual. I know it was short, but hopefully enough to recharge and start. The next presentation will be from Alex Merikli, and he will be talking about a different perspective of the surgical site infection from a surgeon perspective and from what we call the ward. So Alex, to you.
Thank you. Thank you, Yuri. I'd like to thank Mr. Tovey, Dr. Love, Dr. Martina for inviting me to participate in your Capital Day today, and hopefully give you some more information about surgical site infections as they pertain to breast reconstruction and why this is particularly important for XF73. These are my disclosures. One of the most important things that I'd like to convey to you today is that surgical site infections in breast reconstruction are a long-lasting problem. The effects linger with the patient for many, many years, and have an impact not only medically but also on their quality of life. And so this is not just a one-time cost or a one-time trauma, but one that we're learning really lasts for quite a while. There's two main types of breast reconstruction, one that requires a device such as a tissue expander or an implant, and another that uses the patient's own tissue, often from the abdomen, as you see here. During today's meeting, I'll be speaking specifically about device-based breast reconstruction because not only is it the most common method of breast reconstruction in the United States and in the UK and most of Europe, but because it also has an unacceptably high rate of infection. In the US, a device-based breast reconstruction typically starts with the placement of a tissue expander, which is done often at the same time as the mastectomy. And then the patient undergoes a series of fills in clinic, followed by a second outpatient procedure to exchange the tissue expander for the implant, which is usually done about 3 months after the mastectomy. 1 in 8 women will develop breast reconstruction or breast cancer during their life with a total lifetime risk of 12.5%. Currently in the US, there are 3.8 million breast cancer survivors, and the world sees an annual incidence of breast cancer at approximately 2.6 million cases, with the UK seeing 65,000 and the US seeing approximately 338,000 cases annually. Of these women, 30 to 40% will require a mastectomy as a component of their treatment. Thanks to improvements in breast cancer awareness and breast reconstruction awareness, more and more patients are opting to undergo reconstruction. Amongst the women who will receive a mastectomy, 65% will have reconstruction, and this number is growing every year as awareness continues to grow. And the most common method of reconstruction, as I mentioned, is device-based, either using an implant or a tissue expander, which represents 75 to 80% of the cases. This volume, combined with the cosmetic breast augmentation market, creates a market size of $2.8 billion with a compound annual growth rate of 7.2%. Infection in association with breast reconstruction is devastating. It's really far too common, and it's overwhelming, as these multiple images are meant to convey. Infection typically occurs within the first 2 weeks after a mastectomy and device placement and is signified by redness or erythema, pain, swelling, and drainage. These infections must be dealt with swiftly and aggressively, lest the bacteria have an opportunity to create a biofilm around the implant. And if that's permitted to occur, almost always the device will need to be removed and the patient will lose their reconstruction. The 5 largest, most rigorous, and most recent studies on device-based breast reconstruction include a combined total of 12,543 implant-based breast reconstructions. Adjusting by study size results in an aggregate surgical site infection rate of 13.5% for women undergoing this particular surgery, a reoperation rate of 16.4%, and a reconstruction failure, i.e. removal of the device, rate of 7.1%. This is a devastating and far too common complication. Breast reconstruction patients frequently possess a number of unique comorbidities that have been demonstrated to place them at a higher risk for breast device infection. These can easily be separated into disease or therapeutic factors and patient factors. You can see here radiotherapy confers an odds ratio or increased relative risk of 2.8. And among the disease or therapeutic factors that could be considered modifiable are, the only one really is mastectomy skin flap necrosis, which is surgical technique dependent. Looking at patient factors, smoking and obesity would be considered modifiable risk factors that are associated with infection. And the highest risk factor here is diabetes, which creates a almost 4 times greater rate of infection, which is not necessarily surprising. Here I've diagrammed a typical reconstructive course for a patient without complications, which is on the top, and a patient who has a reconstruction that experiences an infection. In the uncomplicated patient, you can see that her entire reconstruction was completed at 6 months without any oncologic treatment delay. To contrast, the patient at the bottom developed an infection at 2 weeks, which was signified by skin flap erythema or redness. This was treated with oral antibiotics. The patient then returned, or the infection returned at 4 weeks and was much more severe, requiring readmission to the hospital for IV antibiotics. And due to failure with improving on the IV antibiotics, subsequently required a return to the operating room for removal of the device. This unfortunately resulted in a delay of her adjuvant radiation therapy and chemotherapy, which may have negative implications on her overall oncologic outcome. As you can see, the device was removed here in the picture, and then a second attempt at reconstruction was started 12 months later. Due to the scarring and lack of tissue that occurred because the infection happened and there was significant skin shrinkage, the patient was no longer a candidate for device-based breast reconstruction and therefore had to have tissue transferred from her abdomen in order to recreate the breast mound. So you can see here, both patients required a mastectomy followed by radiation therapy. The one that did not have an infection was completed with her reconstruction at 6 months, whereas the one that did have an infection, her entire course was prolonged by 15 months. So needless to say, breast reconstruction infection is costly, both in terms of time and money. Here, just illustrating that, 15 months for the reconstruction timeline if a patient has an infection versus 6 months with no infection. And in terms of cost, this infection was directly responsible for a 250% increase in cost, equating to an additional $57,000. And the dollar values you see here are estimates based on this patient's actual costs, which were obtained by the Centers for Medicare and Medicaid Services 2020 cost scale. These costs would likely be significantly more with a private insurer. The negative effects of breast reconstruction are long-lasting. Through the papers you see on the right, my group has studied the long-term impact that device loss has on future reconstructive outcomes. We found that, perhaps not unsurprisingly, the risk of device infection and failure increases with subsequent attempts at reconstruction. For instance, our group's infection rate after the first reconstruction is 9.8%, but this climbs to 33% after the third reconstruction attempt. Similarly, our reconstructive failure rate after the first attempt at reconstruction is 5%, climbing to 22% with a third attempt. Women who experience an infection have a much lower rate of satisfaction with their breasts also, even after later undergoing a successful reconstruction compared to women that had never had an infection. And this is illustrated by the graph at the bottom. You can see here that a woman that had a device-based reconstruction without any complications is highlighted by the bar furthest to the left with the red outline. Whereas if the patient had initially a failed reconstruction followed by a successful one, which is the bar in the middle, their long-term satisfaction with their breasts is significantly lower. Similarly, this study out of Duke demonstrated that even years later, breast reconstruction patients that experienced an infection had significantly lower patient-reported outcome scores for satisfaction with breasts, sexual well-being, and also had a higher level of decision regret. This is an interesting paper that suggests that a breast infection may have a more permanent negative effect on satisfaction and a woman's quality of life, lingering for up to 5 to 10 years after the event. In this paper, at 5 years after their infection, women still had lower physical well-being scores, still had lower satisfaction with their breasts, and still had greater depression compared to women with an uncomplicated reconstruction. Interestingly, despite ultimately undergoing a successful secondary reconstruction, these patients tended to be less satisfied with their outcome as more time elapsed since their initial implant loss. As I alluded to earlier, breast device infection has a negative impact on long-term surgical outcomes, patient-reported outcomes, and of course, cost. The mean cost of suffering a breast device infection is estimated to be nearly $17,000. Additional potential costs associated with the device infection include the cost of readmission, the cost of IV antibiotics, the cost of possibly returning to the operating room for device removal, and of course, the cost for a second or maybe even a third attempt at reconstruction. As is true for many things in medicine, prevention is a much more efficient and likely much more effective strategy than cure. And this is also true for breast reconstruction infections. There are a number of consensus guidelines that have been published illustrating the best practices for a device-based breast reconstruction. These include the use of prophylactic IV antibiotics on induction, antibacterial showering prior to surgery, superlative sterile technique focusing on minimal touching of the implant and copious intraoperative irrigation with antibiotic solutions, and the use of surgical drains postoperatively. An effective agent to decolonize the nasal airways would be a welcome addition to our collection of efforts designed to prevent device infections in breast reconstruction. In conclusion, device infections occur in 10 to 35% of breast reconstructions and have significant consequences. 50% of these infections require admission for antibiotics, and 30 to 50% of the patients that are admitted require device removal, which we've termed reconstructive failure. This is costly not only to the patient and her quality of life, but also to the economy. And it also has long-lasting negative effects on the patient's self-perception. Just a quick back of the napkin calculation that I just did noted that in the US alone, the cost of surgical site infections related to breast reconstruction were in aggregate anywhere between $200 million to $900 million. And that's just with breast reconstruction alone. So optimizing methods to prevent infection really is the key strategy for breast reconstruction. Thank you for your time.
Thank you, Alex. Perfect. Well, a little bit ahead of schedule now. Thanks for that. And maybe we can shift to Bill Love, who will speak about XF drugs and the platform and where it's going. Bill, to you.
That's great. Hi, hi everybody. Can you hear me okay? Excellent. So I'm delighted to present to you XF drugs beyond antibiotics. Could I have the first slide, please? So in this overview presentation, I'll take you through the XF drug platform and why really this is a step beyond antibiotics. And that really is based on the fact that the XF drugs are a new drug class. And this is based on a novel mechanism of action. And this mechanism of action, I hope to be able to demonstrate to you, delivers a number of key new features which make them distinctive from antibiotics, but also allow them to tackle AMR, at least in a number of particular areas that are related to our XF drug pipeline, which will be the final section that I'll take you through. And during this short journey, I'll also be introducing some new landmark data, some of which was published just this morning. So perhaps we can move to the next slide. Thank you. So the XF drug platform, if we look on the right-hand side of the top slide here, you can see that the proprietary structure is based on a central core spacer, porphyrin ring and phenol ring structure, and then on either side, twin cationic arms, so positively charged molecular arms. And it is the molecular variations within this construct that sit at the core of our proprietary patent 3-tier of patent coverage, so composition of matter, mechanism of action, as well as biofilm activity. So in essence, any molecular structures that fall within that type of design, we have our patents cover. And as you'll probably be aware today, we have generated 73 different constructs, of which XF73 is our lead compound. But indeed, we have coverage for many more XF structures in terms of looking for new constructs. XF73 on the left-hand side is the lead compound. It has an international name now, hexaporphinium chloride. But in essence, whatever the structure, all of the XFs that we have created to date have 4 key attributes, which really distinguishes them from antibiotic action. So that's the rapidity of kill of bacteria, the fact that there seems to be a low propensity for bacteria to become resistant to XF drug action, the ability to kill bacteria not only in free form, but also when they're within complex and protected biofilms, and the fact that they have broad spectrum activity against gram-positive bacteria, including all staphylococci, which I'll talk about shortly. Can I go to the next slide, please? So simply put, in terms of the mechanism of action, XF73's positively charged cationic arms will interact with the bacterial cell membrane, which contains a high density of negatively charged phospholipids. And as XFs act on the membrane, next slide, please, they cause a rapid change in the permeability of the membrane, leading to a rapid release of intracellular components, which are vital for life, for the bacteria to survive. Next slide, please. And this loss of intracellular components ultra rapidly kills the bacteria within a few minutes. And it is this rapidity of bacterial kill, which is one of the features which really sits at the core and how it differentiates, or XF drugs as a platform differentiate from traditional antibiotic approaches. Could I move to the next slide, please? So we published this paper today, or we announced the publication of this paper in Frontiers in Cellular and Infection Microbiology. So this, in this study, XF73 was examined for activity against 2,527 staphylococcal strains from around the world. The paper established that XF73 was potent against every single one of those isolates, and all of those isolates came from real patient infections collected from around the globe from 33 countries. It also included 840 MRSA isolates, but also 22 other sort of isolates that were resistant to 22 different antibiotics. So collectively, this landmark study for us has provided data that demonstrates that XF73 is potent against all tested global staphylococcal isolates that we have tested today. And collectively, it also will give regulators, hospitals, that confidence that all patients who could be successfully treated with XF73. So we're delighted with this result. It is a landmark for us and for XF73, and we're delighted that it's been published today. Could I move to the next slide? So I mentioned the speed of action as one of the features that discriminates XFs from traditional antibiotic approaches. And I think this kill curve really demonstrates that feature very nicely. It shows activity against Staph aureus in culture and the impact of drug action and drug kill over time. We see two comparator antibiotics, tetracycline and daptomycin, are only starting to kill bacteria after about an hour. However, remarkably with XF73, we see that 99.99% of those bacteria are killed within 5 minutes of exposure to XF73. And in fact, there are no surviving bacteria after 15 minutes. And so this is a really exciting feature and one of the 4 fundamental features of XF drugs. If I could move to the next slide, please. And really, we believe that it's this rapidity of bacterial kill which then leads and delivers this second feature of the XF drug platform. And this graphic here is a measurement of MRSA's ability to become resistant to a topical antibiotic, mupirocin in this case, but also examining XF73 in the same study. Essentially, anything on this graph that increases shows that MRSA has mutated to become resistant and to throw up resistant strains to the test drug. As you can see with mupirocin, in a very rapid time, probably after about 2 or 3 exposures, you see that there is a mutation and a significant increase in the amount of mupirocin required to be active against that MRSA strain. And after repeat exposures, up to 55 repeat exposures, you can see that the amount of mupirocin required to have activity against this strain of MRSA has gone off the scale. An absolutely massive dose of drug is required to have any impact. Whereas remarkably, with XF73, which is blue triangles at the bottom, you can see that there is no significant increase in MRSA resistance to XF73. So this really, and this is a feature that goes across the XF platform, and it really speaks to a low propensity for the mechanism of action to generate or to elicit the ability for bacteria like MRSA to become resistant to XF73 drug action. And of course, a consequence of this is that we believe that XF73 and other XF drugs could be used in a widespread manner with a lower possibility of resistance or strains emerging of bacteria that are resistant to their action, which in turn means that these the XF drugs can be used in a widespread fashion and not reserved in the same manner as any new antibiotic would be, and only used in a last resort. Could I move to the next slide, please? So the next feature that is common to the XF drug platform is their ability to have activity against bacteria within a formed biofilm. So the graphic at the top here shows how biofilms form within the body. And so essentially bacteria, once they adhere to a surface, so that could be to a tissue or to a medical implant, then they proliferate and then they generate a protective jelly-like coating within on the outside of this clump of bacteria. And this protective coating forms an impenetrable barrier for antibiotics to reach, and also the bacteria grow slower within biofilms. The combination of these features mean that antibiotics are unable to treat biofilm infections very effectively, but also they are sort of a hothouse for generation of mutational resistance to the antibiotics that are sort of being administered. It's also been recently acknowledged that over 80% of clinical infections, particularly chronic clinical infections of bacteria, have a biofilm involvement. So then moving to the data and the comparison here between mupirocin and XF73 from a publication that we have made shows, first of all, the activity of mupirocin and XF73 against the free-floating or planktonic form of the bacteria. And as you can see, they are both potent against the bacteria in that free format. However, when you then look at the amount of drug that is required to have an impact against Staph aureus, in this case within a biofilm format, then you move to the next column, which is the MBEC, or the minimum biofilm eradication concentration. You can see that the amount of mupirocin required to have any impact on the biofilm has risen over 2,000-fold, and so a massive dose is required. Whereas with XF73, in contrast, there is no significant increase in the concentration of this new novel drug to tackle bacterial biofilms. Could I move to the next slide, please? So, so that data that I've just presented over the last few slides is in vitro data. And then I think this next slide really sort of then embodies, well, what sort of an impact can an XF drug have in a real life infection within a living organism in comparison to a traditional antibiotic. And this data was generated by one of our partners, China Medical Systems. It was published about a month or so ago. And in this study, there was it involved looking at an in vivo model of MRSA skin infection. And that skin infection was allowed to develop within a wound, and then either received no treatment, so a non-treated control, received a topical treatment with a marketed mupirocin cream, dermal cream, or with our newly developed XF73 dermal formulation. And the graphic shows the number of bacteria on a log scale of MRSA within the wound tissue. And it amply demonstrates that XF73 was more than 100-fold more potent than mupirocin in killing MRSA. And in fact, compared to the control, there was a 1,000-fold reduction in the amount of bacteria, and over 100 times greater impact compared to mupirocin, highly statistically significant. The other amazing fact from this was that, in fact, this massive difference in clinical impact of XF73 over mupirocin was delivered with a 10 times lower concentration than mupirocin. So I think you start to get a feel of how these attributes from XF drugs and XF73 can translate into impact clinically within the pipeline projects and that we're developing for XF73 in both our nasal indication, but also our dermal indications as well. If I could perhaps move to the next slide, please. So pulling that all together in terms of just a head-to-head comparison against one of the world's most commonly used topical antibiotics, mupirocin, and to contrast how XF drugs, and in particular XF73, can perform against this leading antibiotic. Now, referencing our nasal formulation and the dosing regimen for that, XF73 can affect a very significant decolonization of nasal Staph aureus in a single day, or within 24 hours of dosing. Contrast that with 5 days that is required for mupirocin. The publication that we published today has, again, further increased confidence that XF73 can tackle all strains of Staph aureus, including mupirocin-resistant strains, in fact, any type of antibiotic-resistant strains. Of course, mupirocin is not active against all Staph aureus strains. I've also hopefully given you a flavour and demonstrated the low propensity for resistance emergence for XF73, whereas Professor Wilcox's presentation, you will recall, he discussed the growing issue of mupirocin resistance for the use of that particular antibiotic for decolonization of Staph aureus prior to surgery. In addition, I've demonstrated that XF73 is active against staphylococcal biofilms, whereas mupirocin really can't tackle biofilms, and that's important not only for in the context of nasal decolonization, but of course in our other indication and product development for treating infected open wound infections where biofilms are highly involved. XF73 is really very well tolerated. Mupirocin isn't well tolerated and has poorer compliance. And that's fine in terms of infection efficacy. I would hope that the in vivo skin infection model data that I just presented really does show that superiority of XF73 over one of the world's most commonly used topical antibiotics. If I could move to the next slide, please. So in this final section, I wanted to take you through the XF drug portion of our pipeline. Of course, we also, as Chris mentioned, we have microbiome-based product developments in NTCM3 and also Sporkov, but I'm going to focus purely on the developments that are ongoing with XF73 nasal, looking to enter phase 3 development, XF73 dermal, where we are developing a dermal product for the treatment of antibiotic-resistant infected wounds, and our partners, China Medical Systems, are developing the dermal formulation for the treatment of superficial staphylococcal skin infections. And I will touch on some brand new data within the XF drugs activity against biofilms right at the end. So if I could move to the next slide, please. So this slide really just summarizes the outstanding results that were delivered from our phase 2b study. This again has been recently published, Mangino et al. Earlier this year. But in terms of the clinical efficacy for XF73 was tested in cardiac surgery patients who were nasal carriers of nasal carriers of Staph aureus, and they were either treated with XF73 or a placebo formulation. 73 treated patients saw a reduction in nasal burden of Staph aureus by 99.5%, and this was highly statistically significantly better compared to placebo, with an enormously small p-value. 84% of those patients, XF73 treated patients, had complete decolonization or experienced a 99% reduction in nasal carriage after only just 3 applications within 24 hours. This decolonization effect with XF73 was seen to last throughout surgery and up to at least 6 days post-surgery. This study also demonstrated that systemic antibiotics alone had no impact on nasal carriage over the period of surgery. But it also importantly demonstrated in a clinical patient population that XF73 nasal gel was safe and well tolerated with no nasal findings. And within the treatment period and follow-up period of 3 months, there were no post-surgical site infection that were detected by either treatment arm. If I could move to the next slide, please. So this data and these outcomes, clinical outcomes, are being scrutinized by an expert panel of infectious disease physicians and surgeons And we asked for their thoughts about how XF73 nasal could differentiate from current standard of care, which in many hospitals will avoid is mupirocin. And they came up with that the unique and short dosing regimen of under 24 hours can enable a more greater flexibility in scheduling for surgeries. They also said that it would be much more appropriate for treatment of emergent surgeries and where surgeries had to be rescheduled, or routine surgeries rescheduled at short notice. And also that could be used for treatment of patients who maybe had failed other eradication strategies and agents. And due to the short dosing regimen, they certainly believe that there will be a significantly higher compliance with XF73 having to be dosed for just the 24 hours prior to surgery, as opposed to mupirocin. And recent publications on the compliance with the mupirocin 5-day dosing regimen puts it at about 50%, so just about only 50% compliance with that type of longer-term decolonization that's required with that product. And they also really like the very favorable tolerability and safety profile of XF73. So moving to the next slide, please. So I'm going to briefly talk about the XF73 dermal product development that we have. There's a copy of the previous slide that I described, the tremendous impact of 73 and superiority over mupirocin. I'm not going to go through that again, but what I'm going to do is to just outline that the dermal program development has been moved forward by significant contribution of non-dilutive funding from the UK-China AMR grant award, but also from the US National Institute of Allergy and Infectious Disease, which in combination has provided, I think actually it's about £2.2 million of investment that we've deployed into developing XF73 dermal formulation. And we have now successfully created an XF73 dermal formulation that is specifically for dermal use on the skin. It is distinct from the nasal formulation in a number of ways. And also that funding has allowed us to conduct a large clinically enabling dermal safety study, and that's specifically run by US NIAID, and that study is ongoing, and we're looking forward to presenting the results of that at the end of this year. As I mentioned previously, Destiny, we're developing XF73 dermal for the treatment of antibiotic-resistant infected wounds, for example, for diabetic foot infection. Our partners in China, CMS, are developing the dermal formulation in China for the treatment of superficial staphylococcal skin infections. But just to underline from the data that we've generated in vivo and in the MRSA skin infection model, XF73 dermal gel efficacy is vastly superior to mupirocin. And of course, that gives us and the people that we're working with a lot of confidence, not only in terms of the translation of that within our clinical data for our nasal program, but also more specifically for our dermal product development as well. If I could perhaps move to the next slide, please. So I'm just going to wrap up in terms of presentation of data with, again, a very recent publication in Frontiers in Fungal Biology. And this is the demonstration of XF drug antifungal activity. So in this study, we looked at Candida albicans, which is responsible for a range of infections of the mainly of the skin and mucosal surfaces within the body, such as oral and viral Candida infections. And we looked at the impact of XF drugs. In fact, we looked at 3 XF drugs, and direct you to the publication to look at the detail. The table at the top looks at the planktonic MIC activity, so the concentration of XF drug required to kill Candida albicans, or 6 clinically important strains of Candida albicans in the free form. We get potent activity at a range of 0.25 to 2 milligrams per milliliter. But then we also moved on in this study to look at the impact of XF drugs against Candida that had formed a biofilm. And just as bacteria can form biofilms, fungi can also form biofilms. And again, they are equally important in clinical manifestation and chronic fungal infections. And the graphic or the photos at the bottom on the right-hand side of this slide, in image A, shows fungal Candida biofilm, which is viable. Green staining demonstrates viability of the Candida biofilm. And graphic A is for the untreated control. The graphic B shows XF drug-treated Candida biofilm, and a red translates to dead Candida albicans within the biofilm format. And so we're really pleased to see this antifungal activity, both within the free form, but also within the biofilm format as well. And we're exploring, this is an earlier stage, it's a new observation for us. And so we're exploring the opportunities that are presented by dermal fungal infections with the possibility of development of XF drugs towards products to treat those type of infections. So if I can perhaps move to the next slide, which is my final slide, and just a wrap up really of taking you through that very brief summary and update on the XF drug platform. But the platform is a new class of antimicrobial agent, which has a unique and novel mechanism of action. It's significantly differentiated and strongly positioned against versus antibiotics, including mupirocin, by these 4 key features of the platform: rapid kill, low propensity for resistance emergence, active against existing antibiotic resistant strains, and activity against biofilms, both bacterial and fungal. Our XF drug pipeline of infection prevention treatment products tackle AMR, address high unmet clinical need. Because of these features, XF products are likely to be able to be used in a widespread manner and represent significant market opportunities. And finally, that's to remind that the XF drug platform is capable of generating additional XF family drugs, which could further expand the portfolio and indications that we can develop. So thank you for your attention, and I'll end there. Thank you.
Thank you, Bill. Very, very comprehensive and a lot of potential beyond the XF73. Perhaps we are a little bit over the scheduled time, and we should move to the closing remarks. Before we do so, I wanted to ask if Richard Proctor, Professor Proctor, is still online, and if he would like to add something to all that he heard today as the S. aureus expert.
Thank you. Yes, just a couple of comments. The first is that both in breast surgery and in prosthetic joint surgery, the first infection is bad, but the repeat infection is where the disaster is. And we see that a 5-year mortality after prosthetic joint surgery is after knee surgery 26% and after hip surgery 33%. So if you get infected, you have a very high risk of dying, which is a worse mortality than 80% of all cancers. So prevention is absolutely key. Second comment comes on the heels of what Dr. Bill Love just said about Canada. What's fascinating is that there's a large literature actually about interactions between Candida and Staph aureus, and they cause synergistic disease to be more virulent. And so I think that in particular, I could see the value in the diabetic foot ulcers, where XF73 might really shine. It gets a biofilm, it gets Candida and Staph aureus in a biofilm, and those are major players in this really common infection. So I look forward to seeing how that works out. Finally, we heard a bit about resistance development, and I go back to walking by Bill's poster several years ago, I won't say how long ago, and I saw the incredible rapid killing that XF73 had against Staph aureus. Well, I think that I'll quote Bill Craig, who's deceased, but was one of the giants in terms of antibiotics. And he said, a dead bacterium doesn't become resistant. And I think that was a very profound comment decades ago, and that this is really important. And what we're seeing is that there was just a paper published about a study done in Africa looking at MRSA strains. And they found that mupirocin resistance had developed in Kenya. The dramatic part is that there's no mupirocin available or used in Kenya. So the idea that this mupirocin resistance will only develop where you have the drug, as it turns out, there were strains that carried other antibiotic resistances, and they carried the mupirocin resistance along. And this has become a very large problem in Kenya, but it just shows how resistance can develop. And when I look at 55 passages and no resistance, this is absolutely unique among antibiotics. And so I think the very rapid kill, incredible activity XF73 has, will put it in good stead and keep it working for many years as we lose mupirocin. I don't want to take up too much time, so I'll stop there. But I think that XF73 is the real deal.
Thank you, Richard. So back to Chris for closing remarks, and then we can open for Q&A.
Thank you, Yuri. So I'm going to do a few closing remarks, but after that, we'll open up for the room questions, and then we've got a number of questions online. So prepare if people want to think about. I can see Bill's face getting. If people want to think about questions, I'll come back to you in a second. So, and thank you, Professor Proctor, for those final comments. And thank you to all the speakers today. Really appreciated that. It's obviously an intense session when you sit and watch slides for 2 hours. But I think very clearly we heard about the severity and the very real impact of post-surgical infections on patients. We even talked, the significant mortality risk. And we heard about the strategies for prevention, and we heard about how those strategies for prevention can make quite a big difference in terms of reducing the incidence of post-surgical infection. We saw some graphic photographs of the impact on patients. And I think we heard from physicians who are passionately engaged in this space and who all, I think, are championing an active, nasal decolonization strategy. And we heard some very positive comments from some of our speakers, other than Bill, about XF73. But we heard from Bill, I hope, sort of one of the clearest summaries of the opportunity for XF73 nasal. I think we heard very clearly about the differentiation versus standard of care. And we heard very clearly about Bill's 4 relevant points about XF73, which make it, I think, we believe, such an exciting, new medicine. Obviously, we need to work our way through phase 3 and approval, but the clinical isolate data published last week and announced today is incredibly suggestive that XF73 is going to have utility across the world. I think there were 33 countries involved in providing clinical isolates for that. So, you know, this is a Professor Proctor talking about Kenya. This is a medicine that will have genuine global footprint and can make a big difference to post-surgical infection rates in all countries. I think we heard from our American colleagues on the call about the importance of post-surgical infection management and the costs of post-surgical infection, just purely financially to the healthcare system in the US, which I think we believe very strongly. And in the research that we did over the summer, we got a very clear message that payers will be behind a medicine that can reduce the risk of those significant cost overruns for the system where a post-surgical infection occurs. So I hope that we got some very clear sort of messages to everybody on the call. Thank you for joining, and for everybody in the room. I think what we wanted to do was just be clear about what we believed was the problem and what we believe is the solution, XF73 nasal. And I think Bill ended with a view on what we believe is the future pipeline, XF73 dermal. We heard some suggested directions that we could go with that. We know we need to wait for the end of the sort of the clinical enabling work that's going on at the moment. And we should get those results at the end of the year. And then we need to decide where we take XF73 dermal. There's a lot of different places we can go with that potential medicine in due course. And we also heard about very early stage, but no less very exciting data on the fungal activity of the XF platform. So I hope pretty good session for you. Thank you for the 2 hours. Maybe we can take some questions now, Mark. Bill.
Yeah, can I quickly ask one commercial question and then one about the physician use? If you look at the market research that you've conducted and the feedback from physicians, and I think there's two key attributes to XF73. One is the short dosing window. The other is the resistance profile. I know they're interlinked to an extent, but can you tease out which of those two was more relevant or important from a commercial perspective?
Thanks, Brian. I think it's quite difficult. I think that the feedback essentially was, in totality, what they liked was the profile of XF73. And I think it's still quite small. It's still quite small numbers. I think that what was interesting, though, was that normally when you do this sort of research, physicians get excited about efficacy or safety. What was fascinating here was they were excited about that, yes, but could immediately see a very practical opportunity to improve their surgical efficiency. And I think that was probably more of a surprise to us than we thought, and actually gives another feature, another benefit that we'll be able to use in the clinical differentiation when it comes to taking on standard of care, and also convincing payers and physicians to look to prescribe XF73 nasal. So that's a terrible answer to your question, Brian, because it wasn't totally clear. I think that what we were surprised was that we felt like we had another feature and benefit on the list that would be potent when we were going out to commercialize XF73 nasal.
Maybe to add to that, I think they, like you say, they interplay with each other. I think the bottom line of the interplay of the two really is that you can use this for any type of surgery at any moment and with any type of strain, well, at least known, currently tested and known strain. So the potential is really given by both. It's not only the resistance profile, but it's also the flexibility. To use the word of one of the physicians that was in our scientific advisory board, this has the potential to free up your operating room. because you don't have anymore to reschedule, to juggle around the teams. And if you look at that from a US perspective, for hospitals which live on the throughput of those operating rooms and which have a series of economic implications to that, this is huge, has a huge impact potential.
Yeah. And you think about the UK at the moment specifically, with strikes and all of the issues here around waiting lists. That efficiency is as relevant in a European healthcare setting as it is actually in a US healthcare setting. And I think that one of the things we heard earlier on from one of the speakers was the risk comparison of post-surgical infection elective surgery versus sort of non-elective surgery, the emergency surgery. So I think, definitely that ability to be able to nasally decolonize at very short notice is actually going to be a really important feature.
The second question was a bit more general, actually, was in terms of, you mentioned disinfectants and povidone iodine. I mean, Mupiracin's been around for a very long time, not formally approved in the US for nasal decolonization. Well, why haven't we seen more antibiotics developed for nasal decolonization, do you think?
So I'm not sure I fully understood the question, but so mupirocin is in the US for eradication of Staphylococcus aureus from the nose. It's not for prevention of surgical site infection. Similar situation in Europe is a little bit more patched. In some countries, it is actually for the prevention of certain complications, but mainly also for eradication of Staphylococcus aureus from the nerves. Mupirocin has been proven superior to the other antiseptic. XF73 is superior, or at least comparable or superior to mupirocin if our data is confirmed.
There have been antibiotics that have been tried to be developed for nasal decolonization. I think neomycin was tried.
There are no other antibiotics that have been developed for nasal decolonization that I'm aware of. Yeah. Probably mupirocin is the only one that I'm aware of. Maybe the other experts can add to that. Maybe they know more than me on that.
I'm going to take a liberty here and say, thank you, Brian. The reality of there only being mupirocin out there really is obviously creates a massive playing field opportunity for XF73 nasal when it arrives. Can we take some more questions? Thank you, Brian. Can we take some more questions from the room? Are there any more questions in the room, or should we?
Hi, Andy Smith from Equity Development. Perhaps another unanswerable question from you.
Please don't ask it then, Andy.
Because it relates to what your partner would decide on XF73, the phase 3 study. But I'm sensing you're moving in the direction of reconstructive breast surgery as the phase 3 study population, because it's large, right? And you'll pick up infections and the prevention of infections on such a large study. But also, you talked today about orthopedic surgery and the high risk and the implications of if one of those goes wrong and gets an infection. Is there an interplay between the two? Is it both study populations, or is it breast cancer or orthopedic surgery?
Yeah. So in high level, we are looking at a couple of model of surgeries for our pivotal phases. Clearly, it will depend also from the partnering, but it goes into the direction of the unmet medical needs which are out there.
And I think the reality is, remember, even if it's not the first indication, I think there's an expectation that this medicine will be explored in a number of different surgical types, either through randomized controlled trials or through investigator-initiated studies in due course, as is often the case. The amazing thing for us here is we've seen some of those tables with surgeries in. They are all opportunities for XF73 nasal to make a difference. I'm really conscious of the time. Is there any last question from the room, or we'll take one or two questions from the...
Yeah, maybe connected to what we just discussed, there is a question that was posted online. Is it reasonable to say that XF73 nasal could be the standard of care for virtually every operation in the future? And to connect Yes, absolutely. That's what our market research also suggests. This could be the new standard of care. I think it's beyond the surgery. We have heard the dermal, but of course, even in the nasal formulation, you can think about ICU patients, you can think about dialysis patients, you can think of a series of additional AR and ARD, so that's acute radiation dermatitis. So a series of other indications which are chronic and which are beyond the prevention of surgical site infection.
Okay, Chris, Yuri, as you know, you've got quite a lot of questions that have come in throughout today's presentation, so perhaps we can present those to you after today's meeting, and we'll publish those responses on the Investor Meet Company platform. I know investor feedback will be particularly important to you both, and I'll shortly redirect those on the call to give you their thoughts and expectations, but perhaps if I may, Chris, just ask you for a few closing comments, and then I'll send investors to give you their feedback.
Yes, I think I don't want to sort of repeat the closing comments of a few seconds ago, but I just wanted to say thank you very much to those of you that have joined us here today in the room. It's great to see you. And for those of you that are on the call and have joined the call, or for those of you who are watching this as a recorded call, thank you very much for joining us. Thank you very much for listening to what is obviously a very serious issue for the healthcare system, and one that we believe that we've got an excellent solution for, and we look forward to developing it further and making it available for patients. So, Yuri, thank you very much. Speakers, thank you very much, and thank you, team.
That's great.
Thank you, everyone.
Chris, Yuri, and to our presenters both here in the UK and the US, thank you very much indeed for updating investors this afternoon. Could I please ask investors not to close this session, as we'll now automatically redirect you for the opportunity to provide your feedback in order the company can better understand your views and expectations. So it'll take a few moments to complete, but I'm sure it'll be greatly valued by the company. On behalf of Destiny Pharma PLC, we'd like to thank you for attending today's Capital Markets Day, and good afternoon to you all.